Hi, my name is Simon Wilker and I'm a postdoc in the group Analysis and Redesign of Biological Networks, headed by Steffen Klamt at the Max Planck Institute in Magdeburg, Germany. And in our group, we are working on optimizing microbial cell factories to produce certain compounds of interest. And we do that by applying computational methods, but also methods from the wet lab. And in this short video, I just want to show you how we're using the GNPS dashboard to analyze our mass spec data. As I said, we're trying to optimize microbial cell factories. Usually we're working with E. coli, and in this particular example, I just want to show you we're engineered E. coli to produce the biofuel isobutanol from glucose. And here I just want to show you an example where we want to compare two different strains we generated regarding their performance of isobutanol production. So if they have a higher titer or yield and productivity. The nice thing about isobutanol is that it's a volatile compound and can be directly quantified from the culture broth by a GCMS, by headspace injection. Thing is, in our lab, we only own one GCMS instrument and also only one mass hunter license for this instrument. So sometimes there's a little traffic jam in front of the machine because people want to analyze their data or want to generate new data. That's why I'm usually analyzing the data not at the PC connected to the mass spec, but at my office PC. And that's why I'm using the GNPS dashboard. So as you can see here, I just downloaded the raw data from the from Mass Hunter, from the GCMS, and to be able to analyze it in the GNPS environment in the dashboard, I usually convert it in just one click with the MS Convert tool into the MZML data format. I already did that. Then to make everything a little bit faster, I uploaded the data I just generated to the GNPS environment, so I can just directly import it into the dashboard, which I did here. Here I just, this example, I just imported the standards, not the samples, the data from the samples of the E. coli strain. You can see here, so I measured them in sim mode, and which was, here very easy is to extract an extracted ion chromatogram, which I use for a quantification. So here, as you can see, I'm extracted the MZ of 56.1, which is a fragment ion of isobutanol generated in the GCMS. And as you can see here, I just did the standard in duplicates, and here's an overlay of all the data measured of the standard. And as you can see here, they fit quite nicely. So the duplicates are laying pretty much exactly on top of each other. Now, which is very nice in the dashboard is that they also automatically integrate the peaks of the extracted chromatograms. And you can just by click on the export button, and then you get a nice Excel table, which I also prepared already with all the peak areas. And so you don't really have to write them down or anything. You just can export them directly in one click. And with this data, I This again is a screenshot of the overlaid iron extracted iron chromatograms, and with this I could generate a really nice calibration curve. And using the formula from this calibration curve, I analyzed the samples from the 2 E. coli strains. As you can see here, the strain 2, the blue one, is performing slightly better than the strain 1. Also, yes, she has a higher titer and also has a higher specific productivity. Yeah, I think that was already it. I hope I could show you that the GNPS dashboard is a very useful tool also for metabolic engineering labs like ours, where we don't use that advanced mass spec methods, but we're just using mass spec for quantifying our compounds of interest and our metabolites. But still, especially if you don't have that many licenses of the original data analysis software, it's a very useful tool to switch from lab PC to office PC or even to home PC to work on the same data or share your data or work on the same data with your colleagues. So I hope you liked the video and thanks for listening. Bye.